Thank you. All right, right, we're underway. And indeed, this talk is pretty much like the biodiscovery effort for drugs from the sea. It started off pretty slowly, but it gathered a lot of pace in the 90s, and then it's dropped off again up until about now. So this is a very timely talk because there's a resurgence of interest in looking for new medicinal leads, particularly for things like COVID from the sea. Before I start, I just wanted to acknowledge a few really very important people. Important to me because these people helped me along my pathway, but they're also famous in their own rights internationally. The late Dame Professor Pat Berquist, she was my supervisor through both of my degrees at Auckland University. She's internationally renowned for her work on sponges, and you'll soon come out of this talk just loving sponges, I can guarantee it. And you'll all want to sign up for our courses in sponge ecology and chemical ecology, but Pat really brought that to life at an international level. And she was probably the leader in this country, along with the chemist colleagues, Murray Munro and John Blunt from University of Canterbury, Con Canby at University of Auckland, who really put Aotearoa New Zealand at the forefront of drug discovery programs from in and around the world, and you'll see why that is. The late Professor Joe Baker, he was my boss for a while and certainly a mentor of mine through my Australian years. He was one of the first marine natural products chemists ever. He made himself famous by discovering Tyrian blue, and that's the pigment that dyes Roman cloaks. It's been known for a very, very long period of time, but Joe was the one who actually worked out the structure of the molecule and its synthesis. Professor Mary Garson, she was my mentor, she was my supervisor through my postdoc years, particularly in Australia. She's a chemist of international repute, as you can see, and is now still working in marine natural products chemistry at the University of Queensland. And Dr. Peter Murphy, he ran the Australian program while we were running the New Zealand program for the New Zealand Australian National Cancer Institute drug discovery effort in marine systems from 1986 to 1996. I'd also like to note Shirley Pomponi. She was director of Harbour Branch Oceanographic. We interacted with them in the US. And in those days, they were funded by Johnson & Johnson. So you see there was this link going back many, many years in the marine space from drug companies into the marine research area. These are our partnerships. These exist still to this day. Internationally, we are very well linked. We have partners in pharmacy. This is a European consortium. The National Cancer Institute, we're very closely aligned with them in the US, and the Scripps Institution of Oceanography in La Jolla. And the three on the right there are relationships we have to China. So the very top one is Yantai, which is the sister city to this city. And their Institute of Coastal Zone Research has a very strong drug discovery program. And SIM stands for the Shanghai Institute of Materia Medica, so there are quite strong links through to China as well, which we intend to use soon. So as I launch into the talk proper, here's something for you to think about as we go on, and there are bits and pieces of clues to resolve this conundrum. There's a linkage between a sponge, a crab, and cancer drug discovery, particularly that compound down there called Halaven. So I'll resolve this for you at the end, but just have a think about how these things might be connected. It's all to do with chemical ecology and what I'm particularly interested in, that is the role of these sort of interesting compounds from the organisms that deliver them in nature. So I'm not sure if many of you have seen this movie. Hands up, who'd seen this movie? Medicine Man? We'll try and run a little bit of it. I show this now because part of the story is about biopiracy, and there's some quite prophetic elements in this movie. It was based on land, of course, that can be mapped into the sea. And all of the elements of research are in it. There's partnership problems, of course, what do you expect when a chap goes out into the jungle all the time. There's been this search for cancer, and then, of course, where does the research funding come from? So it's all in there, and we'll see if we can play a little bit of it, I think, for those who haven't seen it. Oh, there we go. It's not going to play for us, or will it? Here we go. And hopefully the sound will come through. I play this because it does tend to give bit of a perspective of what happened in the biopiracy days.
Robert Campbell has gone deep into the Amazon rainforest, into a world of astonishing beauty, a land of ancient mystery, where he's made an amazing discovery.
What is it you think you've got?
I don't think. I know.
You found the cure for cancer, and all you can say is, I know?
There's just one problem.
You did what?
I've lost it! Haven't you ever lost anything? You pass your car keys? Well, it's rather like that. Now you have it, now you don't.
A miracle without proof is only a miracle.
Now they must rediscover that miracle.
Well, I'll leave it there, but there's a few things from that that you might note. They went into a rainforest, an area of very high biodiversity. There are issues of provenance, of course, because there's a question about their interaction with the local communities there. I'll just get this back again, sorry. Oh. Let me talk to you about another matter. Here we go. Let's flick through that now. Okay, and you might have noticed some of the technology you had, which was enormously well advanced for 1992, because it's only just recently we've got desktop HPLCs and mass specs and things like that with the dot matrix printouts. But it's an interesting story, and you'll see elements in the talk that will come from that movie, and that is going out into areas of high biodiversity, having a look for leads for biomedicines, and then trying to do something with it. And there's all sorts of issues in around that in terms of who owns the IP, what do you do with that biodiversity as you collect it. The story is about actually losing the original source. And it's all around trying to come up with novel medicines that are going to be needed in a planet that is now overpopulated and where we're seeing things like COVID-19 come up more prevalently. So in the past, that search through nature, through natural biodiversity, was called bioprospecting. But that, of course, invoked this idea that you went out and then mined the resource. And indeed, there are many cases, particularly in rainforests and elsewhere, where they did, in fact, mine some quite rare resources, plants, fungi, and the likes, almost to the point where they were lost. We have coined this term biodiscovery because it suggests that we're not going to go and mine the natural resource that's out there. Rather, we're going to come up with other ways of producing either mimics of the molecules or coming up with other ways of producing them. So the talk's all around pharmaceuticals, but there's all sorts of other bioactive leads of use to humankind that we can develop from this discovery program. And I'll give you some examples of what they look like of relevance to this country at the end. So useful biomedicinals, if you like, from nature. You will know these already. Aspirin from the bark of willow trees, digitalis. You can kill people with it, of course, but there is a drug that's used for cardiac issues. And so there's this natural progression of going to the land, looking at plants in particular for medicinal outcomes. This isn't new. Chimpanzees have been biodiscoverers well before we have. And it took a little while for people to realise that where chimps were around, if you watched them often enough, particularly when they were sick, they went to certain plants and then ate those. If they had a profusion of parasites, for instance, in the gut, they'd go to a particular plant and eat it. And so this idea of looking to nature for some clues came out. And the researchers have since, with the indigenous folks of the areas, which is a good thing, have then sped up their discovery programs and looking for drug leads from nature. So that's what we were doing in the sea. So the program that we were involved in from the mid-80s right through to probably the early 2000s was taking this discovery pattern on land back underwater. So our role in that, as part of a very large team, was to look at biodiscovery in this country. There was a parallel program in Australia that I went and was fortunate enough to be engaged with, to create marine biodiversity libraries. And of course, the minute we went into the sea, we found lots and lots of new species. Indeed, that's happening today. Just in diving out in the harbour here, just over the last year, a student of ours has discovered 2 brand new species of sponge. We'd be surprised, new to nature, and he's just recently published on those. So when you look, you find new things, and they are full of interesting compounds. So they, as I said, they have medicinal outcomes, but all sorts of other outcomes as well. If we look to the current sources of modern drugs, we can see that about half of them are directly derived from natural products. So this is from nature. That yellow piece of the pie chart are the synthetics. So these are synthetics, but they're analogues of discoveries made from nature. So most of the leads that we have for our drug therapies have come from nature in some form or other. That little core there, the blue core, represents the leads that have been successfully developed from just pure chemistry. They call that combinatorial chemistry. So that's where the medicinal lead has been invented by chemists based on what they understand of other chemistry that seems to work. It's very small, and they haven't had any major discoveries in that space for many years, despite expending quite a bit of money. So we look to nature and we can find the leads. And the reason for that is that with diversity in nature, you have a lot of diversity in the chemistry. And you have systems that are going on where there's a lot of chemical interactions, particularly amongst the animals, that we all share very similar metabolic pathways. And so the chemistry that works in other animals will potentially work on us as well. So you get a much higher chance, even from random survey programs, of finding something of interest. If we look at this diagram, don't worry about the small print. The stuff along the bottom, the print along the bottom of that graph, the various phyla, animal phyla in the animal kingdom, and the categories moving into the 3 dimensions there are the different, ecosystems on the planet, including tropical rainforests. And you can see that the bars, all those red bars, that refers to the phyla on the planet that come from the sea, okay, on the seabed. So almost every single phylogenetic area is populated by organisms from the marine seafloor, much more so than you can see from even rainforests, although the insects tend to predominate there. So from the sea, 90% of the known phyla are represented, and therefore that equates to a really high diversity of chemicals, bioactive chemicals, as you'll see. So this is a representation of a cell metabolic pathway, and each one of these purple names refers to a lead from a marine source that will interfere with that cell if it's not functioning properly. Now, the importance of this is that there are leads for interfering with cell metabolic process at every single stage around that cell cycle. And usually these types of compounds interact with cells that are behaving badly. And of course, what are tumour cells? They are those sorts of cells. They behave abnormally, and these compounds are special in the fact that they can actually interact specifically at those sites. So there's a lot of diversity of choice when you're looking for a therapeutic to decide on where you really want it to upset cell cycle so that the cell can be identified and will be killed by natural immune systems. Another way of looking at why marine leads do exceptionally well, particularly in cancer therapies, is by having a look at this representation of a screening portfolio. So what we did with our relationship with the National Cancer Institute is submitted our samples, our marine samples, to the National Cancer Institute's 60 human tumour cell line. Okay, so it's a preclinical test. They have cell lines of human tumour cells, 60 of them, and they're categorised into various groups: leukemia, lung, colonal cancer, central nervous system, melanoma. The 2 outside bars there are the baphlomycins, and you can see by melanoma they have peaks on the right-hand side of the line there. That's showing that they're active in that area. If you had peaks on the right-hand side of those lines for everything, you might think, well, that's really active, but it's probably too active. You've probably got a really grossly cytotoxic compound. So in a drug discovery program, what they are looking for, what the medicinal chemists are looking for, is activity in just one therapeutic area of interest, because that demonstrates a particular sensitivity, if you like, or specificity of targeted action. And that's what a lot of these marine compounds do. So we have salicylamide A and lobotomide A inside the red square there. One's from a sponge, one's from a soft coral, both from Western Australia in this case. And you can see that they have these sort of peaks in the melanoma area, and indeed that's what those two compounds are being developed for. So not only do they interact with tumour cells, but they interact quite specifically. They're quite selective, which is a good thing. That's what you want. This is just a slide that shows how well these can work. This is Yondalis. It comes from sea squirt, which happens to be an invasive species. We've got a closely related species here in the harbour to this. It looks very similar. That's the molecule, and this is significant probably in this presentation also, which I'll touch on towards the end. These are big molecules, amazing things. How that sea squirt can synthesize, make that molecule is not well known, but they do, and we'll talk about that a little bit. But you can sort of see the problem, of course, in being able to produce it artificially. Anyway, ecdenocidin, or Yondalis, is used for soft tissue carcinoma and ovarian cancer, but it started off its life as a therapeutic in lung cancer. And you can see in this patient, it's the same patient, you can see those white blemishes in the lung area and how they've disappeared. So these things work. And they often work by targeting the tumours, stopping them growing, and so your immune system can come and mop them up. So the side effects are a little bit lower. You get all sorts of other interesting things in the hunt for medicines. Anti-inflammatories. There's a lot of anti-inflammatory compounds from marine organisms. Here's one that I know and love. I've actually tried this. I went out on purpose and bought my mother a pot of this. It's very expensive for Mother's Day. And the reason was that I wanted to try it myself, which is probably not a very nice thing to do to your mother. She said, "This has already been opened." I was like, "Oh, it's all right, Mum. I was just making sure it's okay." Anyway, it works. It works really well. And, you can see that the industry for Estee Lauder's resilience is about $7 billion per year. Once again, that molecule, even though it's a lot more simpler than the previous one you saw, is still quite hard to make, to synthesize. It turns out that little sea whip can make it really easily. And that's now the subject of a sustainable livelihood for peoples up in Ponape and some of the Micronesian islands. They grow it like tea and then snip the things off and extract it. So that's just a little aside on other things. If we get back into the therapeutic area, particularly in the anti-tumour active drug discovery programs, and we have a look at what marine organisms are producing the best leads, we always come back to sponges. This is where I really hook you all now into looking at sponges. POR, the porifera. And you can see both for cytotoxic extracts and for the anti-cancer leads. So these are anti-cancer leads that don't have really concomitant cytotoxicity. The sponges do well. Ascidians are probably the next best, and that's sea squirts. So this is what sponges look like. And one of the reasons that they are so good at providing these leads is because they're so old. Okay, so sponges are the lowest metazoan, that is the lowest animal, multicellular animal. They've been around for about 800 million years. So that's a long time to be able to evolve. And of course they haven't evolved hugely. They still sit on a rock and look fairly boring, although they're usually beautifully coloured. What they've evolved to do, though, to maintain themselves, is they have developed a very sophisticated chemistry. And that's what makes them of strong interest to us. Added to that, they have managed to get on with microbes in the sea. The sea is full of these things, bacteria and viruses. And some of the sponges, that tropical one up there, Rhopaloides, all of those little round dark shapes are bacteria. And it is full of them. This one here, Mycale, it produces a very active anti-tumour lead that's currently in development. And it has specialist cells that actually garden little populations of bugs, of microorganisms. And you can see that B refers to a bacteriocyte. And once again, all of those little dark ringed features in there, this is an electron micrograph, are bacteria. So it's gardening these bacteria, and cells around there, these ones with the dark stained inclusions on them, are called spherulous cells. They're chemical factories. And what we understand is happening is that the sponge is gardening selected microorganisms and then transferring some of the compounds from them into its own metabolic systems in the spherular cells. So really, really tricky stuff. They're also massive filtering machines like no other. So if I had a scattering of sponges, say on the floor here, filled this room up with seawater, they would filter the entire content of the room probably in about 3 or 4 hours. It wouldn't take them long. The exhalant plumes run at 2 knots. Their metabolism is enormous, and they would strip out all of the ultraplankton out of the room pretty much in the same time. So let's have a look at what they might be engaging with, because that's part of the story as well. So we come now into marine microbiology, and marine microbiologists are interesting people. They like counting things, I've found out. The Sogan's team worked out there are 38,000 microbes in 1 litre of seawater, and he wanted to find out how many microbes, these are bacteria primarily, were in the world's oceans. And he reckoned there's about 100 times a billion times a billion times a billion cells in the ocean. And the reason for the elephant there is I wanted to get a feel for just how much that was. And so we converted that into what equivalent elephant weights were if you filtered out all of the bugs out of the world's oceans. An elephant weighs about 4 tonnes. Any ideas of how many? Okay, here we go. 240 billion elephants in weight. And I worked out for Tauranga Moana on a full tide, we're probably dealing with about 2 to 4 elephants in weight of marine microorganisms. But if there was a plankton bloom, it might be double that. So there's a lot of microorganisms in the ocean. Not to be outdone, a chap named Curtis Suttle decided this one works in viruses, marine viruses, and he found that 1 mil of seawater has about 50 million viruses in them. And so he decided to outdo Shogun and virtually put them end to end, and figured that they would go out to space, if you put every single virus in the world's ocean, to about 60 galaxies. Okay, so the sea is a biologically active chemical soup. And so that way we not only have these lowest metazoans, 800 million years or so old, interacting with their own bugs, but they're also constantly coming into contact with things that they're filtering, both good and bad, and they need to react to that. So they live in this chemical soup. And it's from that they're able to do some pretty amazing things. We know that already that there's lateral gene transfer through a lot of these things between marine bacteria and other organisms. So they harness a lot of that chemistry, and they're very flexible in doing that. So they're not hardwired, many of them, into just making one particular compound. They can actually do quite clever things. So these are the drug leads from the sea up until reasonably recently, and I use this table because it's the result of the work that we did in Aotearoa and in Australia. And I popped it up because I also wanted to highlight the ones that we have contributed to. Okay, they're all of the ones in black there. Those are the Australasian leads, and most of them have come from us, from New Zealand, and also the Antarctic. So 30% are from Australasia, a really high hit rate, given that our contribution was actually relatively minor in the global program. Okay, so this is the results from the globe. Some of these things you may well know. The cone shells, which is a compound out there now, it's a drug, Prialt. It's for intractable pain, and of course it comes from cone shell venom. That was developed at the University of Queensland. So why do we have such a high hit rate at finding these anti-tumour active leads? Well, fortunately, we have a census, and it's ongoing, and I was very lucky during my time at AIMS to look after a group that was involved with the coral reef census of marine life. So there's some results from that, about 250,000 species. These are marine animals, okay, marine macroorganisms. They think there's about 1 million, but those are the ones that have been formally identified so far. If we have a look at how we're doing, our EEZ is the 4th largest. It's about 1% of the Earth's surface. If we have 250,000 more or less species worldwide, we have about 17,000. In fact, it's now 17,137 with the 2 new sponges that we have just found off Tauranga Moana. So 14% of the species. So we've got a pretty high diversity. And the other important thing is the endemism, the uniqueness, is very high, as it is on land. our marine environment and the marine plants and animals there are unique to this country. So that means two things. One is there's a lot of diversity to go and sample, but what we find is likely not to have been found elsewhere, and that's important when you're considering the intellectual property space, because you're coming up with new compounds, new species, new compounds, and that can be very important. The other reason is that we were all marine ecologists in this hunt. Elsewhere around the world, and you'll see a little bit more of this, the people who are looking at drug discovery programs were chemists. Now, are there any chemists in the audience? Hopefully not. What I'm about to say. Chemists don't like variation in nature, I've found. Nor do physicists, actually. But nature is enormously variable. And so the drug discovery companies weren't really interested in compounds that had a lot of variation in them, that weren't too stable. To us, they were the really interesting ones because it meant that there was a lot of flexibility going on. And I'll give an explanation of that. We probably, the term chemical ecology has come up because we want to understand why these organisms produce these compounds the way they do and what their role in nature is. And most of it is, when you're dealing with things that need to sit on a rock to live, is defending their real estate, their patch, occupy space and defend themselves from things that want that space. And here's a classic example. You've got this little sea squirt in the middle of there, a didymnum. It produces known highly bioactive compounds, which are also initially in the cancer screening program called didymnums. And you have the soft coral that really wants that territory. But you can see that little Sidian, and there's another one beside it too, Naplidium, it's holding its own. So it's producing these compounds, which this was in a shallow water, so there's a lot of water going over that. So the dilution of anything it exudes is huge. So these compounds are super potent. They clearly are having an effect because that soft coral just isn't wanting to grow anymore. The cells have stopped growing in it. And if you have a look around the border of the soft coral there, there's no death. So the acidity is producing a compound that's not toxic. It's just not killing the cells. It's rather getting into those cells and stopping them from growing. And that, of course, is probably what you want for an anti-tumour active lead. You want something hugely potent, something that can get inside a target cell, and then something that will just stop it because it's aberrant, and then you can have other mechanisms to clean it up. So that's one of the reasons why we were successful, because we went out to places to look for these things. The other thing we found, as I said, is a lot of variation. One species has lots and lots of different configurations on similar molecular themes. There will be a test on this too, by the way, so note where all of those things are. This one, the Discarabdins, Lactranculia, it's found right throughout the southern hemisphere, mainly in colder places. There's about 30 now configurations of that particular molecular theme or moiety. And not all of those constructs are equally active. So this is the family of helicondrins from the lead that has done exceptionally well. It's now a drug from New Zealand. Talk about that a little bit more. And here are there are 9 different types of helicondrin molecule, and most of them only vary with one atom here and there. But it shifts the whole sort of three-dimensional shape of that atom, of the molecule, and that then causes it to interact differently depending on what it's applied to. And you can see here helicondrin B, which was the precursor of the drug Halaven, a late-phase breast cancer active, has an enormously potent activity in the initial targets it was put against, which are melanoma cells, 0.09 nanograms per mil, enormously potent. And yet helicondrin A, one atom difference, has no activity whatsoever. So that's telling us that these sponges or other marine organisms are creating a bit of a family of compounds, or they have a sort of a base molecule that they can stick things onto by harnessing some of the bacteria and stuff in and around them. So they're quite clever. And so they can actually create molecules on the seabed in all of those horrible rough conditions to fit to purpose. And that to me is really exciting. We wanted to check whether that was the case, and we thought, oh, well, the only place to go and really have a look at this theory is to go somewhere where there is no change at all on the seabed and where there is a pristine environment. So we thought, well, let's go down to the Antarctic. We knew some of these things were down there. Flew down. One of the first problems was finding somewhere shallow enough to dive. So you start off by digging a little hole. Then you get a bigger digger and build a bigger hole. Hop down to the bottom. This is a little bit grainy because it was taken with one of those really old Nikons ages ago. But those white posts were sediment traps because at the same time, we were worried about some of the permafrost melt. That's another whole story. And we set up those sediment traps. But the reason I point that out is it's crystal clear, really stable. Those sediment traps are about 150 metres away. It's amazingly clear, near the optical clarity of seawater. But you can see that's a sponge garden. There's as much diversity there as in a coral reef per unit area. It's absolutely packed. And we figured, well, it's so stable down there, it's always minus 1.98 or 99 degrees C, there's no real change. And we tried to dive as much as we could under the permanent sea ice, so it's pitch black. But down there we also found that, yes, if we ran experiments and triggered an immune response or something in these things, they could respond by reconfiguring those molecules into different configurations which were active against the disturbance we're putting on them. And the interesting thing is that they were doing that at -2 at exactly the same rate as the same experiment was done up in plus 18 degrees up here. Now, the biochemistry and the enzyme activity to allow that to happen is just incredible, because to do that synthetically, you need a hot acid bath with all sorts of other reagents in it. So we haven't done this yet, but there is a lot to learn on how these organisms are manipulating really complex molecules into a desired direction. So that's what this is all about, some drug design opportunities. We know we can coerce these things to manufacture or biosynthesize these molecules into a more desirable direction. The next thing is to see whether we can do it into a direction that is more appropriate for humans' use. So it sort of begs the question, though, why not just kill your neighbours? Some of us might like to do that sometimes, but you never know when they're going to fight back. Yep.
Sorry to interrupt you here, Chris, but that diagram in the white square looks very complicated. I'm not a chemist, but who designs that and works out what it all is? Is it you? No. The people who will be able to draw that diagram are chemists themselves. Yeah, absolutely. And it's a massively tricky job. How they do it is using a thing called a nuclear magnetic resonance spectrophotometer, which holds a small sample in an enormously powerful magnetic field. The one we've got, 600 megahertz. There's one in Brisbane which is 900 megahertz. Don't take your cell phone or swipe card with you when you go near it. But they then shake the molecule, and the magnetic field around it allows you to see, I guess, the stresses between the various atoms and molecules in a carbon chain. And from the various stresses in between the different molecules, they can work out the three-dimensional structure of it. Put it there. So, yeah, no, people way, way more clever than I do that bit. It looks very complicated.
Yeah. Well, the thing is, the ones that actually design these things, of course, are the organisms themselves. And for many of these molecules, helicondrin B, the one that generated the Halaven from New Zealand, the drug Halaven, that has been synthesised only once as a PhD project, and the yields were infinitesimally small. So, and I'll get to that story in a minute, actually, on how you actually produce these complex molecules. But it was a good question, because, yeah, we have a lot to pay homage to for the marine natural product chemists who actually work out the three-dimensional structure of these massive molecules. And if you're dealing with things like proteins and things as well, man, it's even more so. So, yeah, the reason why they don't kill their neighbours is because if you do that constantly, you might end up with a neighbour who's got more weaponry than you do. Okay, so that's usually the case. So part of this, a term that sort of springs from all of this, in fact, it was a precursor to it, is a Lilopathy. And this is the interactions are well known in the terrestrial domain.
from interactions between plants. I mean, you often look into a canopy and you can see the plants are not sort of touching one another. They use their chemical defences as well, exactly the same. And in a complex marine environment, allelopathy is happening all the time. So you get different interactions from the same organism depending which border it's engaging with. In some places it's being aggressive, thinks it can take over. In other places it's in a standoff situation. So the chemistry is quite complex. And we have a grant right now from MB looking at allelopathy and the chemical weaponry used, particularly when we're dealing with marine invasives. So here's another element of chemical ecology which hastens our ability to really quickly find neat leads. Because, of course, if you're an invader, a marine invader, be you a soft coral or a sea squirt or something, and you're wanting to stick to a surface which is already occupied by native species, you need to be able to somehow switch off the defensive responses of that native community. And that's where this chemical warfare comes into it. And that's what these marine invaders do. So we had a look at that list that we had up there before of the leads, and I thought, well, let's interrogate that list and see how many of those leads are epizoic species, so things that live on top of other ones, species that need to switch off the rebellious response of the organisms that they're sitting on. And we found that half of them are epizoic species, so half of them will sit on top of another species. Then we looked at that half and found that 87.5 were invasive species. So things that are coming in on ship hulls, things that are in our harbour now, these are aggressive chemical warriors, if you like, and they do well. And of all of those, they all had some form of immunosuppressive activity. So once again, if you think of it from a sea squirt's perspective on coming up to a wharf piling, as a larva or something, it needs to switch off the antagonistic response of things it's settling on. And that's what they do. So this is hopefully just giving a bit of insight into the cleverness of the chemistry and how it's made, and really the sophistication of some of the interactions of the organisms that we have on the seabed. I sort of hinted at this before. I mean, the rainforests of the sea have been argued to be like tropical coral reef systems. And this is where a lot of the world's biodiscovery programs have gone. It's really nice and beautiful to go there. We, however, found much better territory to harvest inside smelly old ports and harbours and systems, and dare I say, near sewerage outfalls. You go to the yucky places. But anyway, we did really well in these places. And we found things like this. Now, this really breaks my heart. It's a very sick, sad sponge, still alive, hanging in there. And it shouldn't look like that at all, but this was near a sewage outfall. But the interesting thing is, it produced these very strongly, the sick one is in blue, really strongly herpes simplex and poliovirus antiviral compounds with no cytotoxicity. So it was producing compounds that were getting into the cells, knocking out the virus, but not actually killing the cells. Now, antivirals are quite rare. That's what it should look like. That's a healthy one, which was taken in the same harbour. This was from Otago Harbour, about 500 metres away. So once again, it just gives you an idea that when these organisms are challenged by even abnormal events, in this case a human-derived sewage plume, they are responding to some irritation, some challenge in their water column very specifically. And this once again gives us a hint as to where we might be able to go into the future by actually using them to design and initially biosynthesize the compounds. So there's a whole bunch of leads, and many of these have yet to be taken up. And as I said, there's now a resurgence of interest in following up on some of these old leads. And one of the biggest problems was how you produce them, particularly at the early stages. Now, this is the drug lead from here, from New Zealand, Halaven. It's now being used as a late-phase breast cancer therapeutic, and is also now going to be joined up with a number of other drugs as a prophylactic for breast cancer. It's doing really well. But how do you produce it when you really have a lead and it's known to be really hard to make, very expensive to make? So there's a bit of a chicken and egg situation here. The drug companies won't want to take this forward until it's proven its worth. You can't prove its worth until you have enough of it to be able to actually take it into the clinic. So that's why a lot of the natural product leads, particularly from the sea, don't go anywhere, because they're constantly competing with synthetic analogues from elsewhere. But there's now, as I say, a resurgence of interest, because synthesis, the ability of chemists to actually make analogues, is now much, much better than ever was. So the other part of this story is an interesting one, in that this compound came from a very rare sponge. We estimated how much there was on the planet. It's about 300 tonnes, plus or minus about 50 tonnes. It's only ever been found where that star is on the lip of the Kaikoura Canyon. That's Kaikoura. And of course, you know about the earthquake. That lip has broken away, so that might have been lost. And this is a story that we're going to head into now, and that is we're losing these leads. In this case, it was through a natural catastrophe, but what we're doing to the sea, from the land in particular, is now causing a loss of massive biodiversity. So we'll get to that. Just before I do, this is the drug discovery process. The discovery phase, the preclinical lead phase, and then phases 1, 2 and 3 in the clinic. There's a fourth phase, which is the big rollout. It costs, for Halaven, it cost $800 million US to get that drug on the market. It took about 15 years. And it was put on the back burner for many, many years because they couldn't get enough. So to the discovery phase, you only need a few micrograms of the compound mix. But to take it any further into the late phase preclinical trials, you need a gram purified compound. And for a sponge, that really means you need a tonne of sponge to generate that gram. Now, most of that's water, but it's a lot. And you can imagine, well, how do you produce that? We don't want to become biopirates. We don't want to go, and we were very tempted to, I might add, because it had huge potential. But we knew because of this particular lead, it was quite rare, and you could do significant damage to the only known population of it if you went and tried to trawl it up. It's quite deep, that one. So what are the other ways of doing it? This is another example of the supply side issues. 847 kilos of moray eel livers will just give you 0.35 megs of ciguatoxin, for instance. So this is where the idea of biopiracy came out, because there were people going and hunting out nudibranchs, these sea hares, eels to just get the livers, and creating molecules out of it. It was unsustainable, and it created quite a lot of history. We got around it by going to aquaculture, and this is where a lot of the sponges were growing, just in aquaculture. So that was the way through. So it was quite a significant lead. Aquaculture has been a big winner, really, for all of the down under leads. We've grown all of these for various outcomes, but it's dead easy. It's a piece of cake. And it demonstrated a way where you could produce these things before they were proven up without harm to the environment, because we collected in a sustainable way, and created that initial mass of a tonne or so to get 1 gram of extract. Worked really well. There are companies now, Pharma for the ectinocidin, for the ondalis, that are now doing this as a regular thing in the Mediterranean on the sea squirt. It works really well. This is some of the colleagues at Scripps and their work on Brystatin A. And Dominic Mendola, a colleague of ours, he's now just retired, sadly, but he worked out that you could actually supply the global demand. We did the same thing for Halaven, simply from aquaculture for that particular drug. So just one of these structures, they're not very big, can service 130 patients per year for that particular therapeutic. It was our calculation for Halaven B, for the globe's population who needed that therapy, was 80 longline farms would service the globe's population. But that won't happen. Drug companies are very nervous about any form of natural production, particularly in the sea. They would rather go to an analogue. And for those big molecules, what they do is they look for the warhead of it, the active bit, and they synthesize that because it's a bit easier. But this led in the earlier days to very bad collections. 22 tonnes of Brystatin, or rather bugula, the bryozoan, the bit of fluffy stuff you see on the walls, were collected for Brystatin A in the early days, 22 tonnes. They scoured every last bit of it out of a significant part of the coastline to find that none of it had any of the activity at all. But it did cause this term to come up, Biopirates. And it means where you go out and you collect without permission in other people's countries and other people's backyards, and where you do damage to the environment in the pursuit of trying to find these drug leads. So there's a lot of discussion and a lot of controversy around biodiscovery, prospecting projects in around the world. So much indeed that the Captain Hook Awards are a regular thing in the Convention of Biological Diversity, and you'll see we actually feature in this particular one, 2006, the Axis of Evil Award, Canada, Australia and New Zealand. And this, we got that not so much for anything to do with biodiscovery, but because our countries were slow off the mark in making a fuss about terminator genes, the Monsanto type things. But because of this and the outrage, there are now quite significant controls and directives from the Convention of Biological Diversity and this a thing called the Nagoya Protocol to, I guess, provide guidelines on safe and effective and sustainable ways of discovering things. So, as I said, aquaculture, fermentation, gene expression and things like that are all opportunities to supply these sorts of organisms. Now, I'm just going to finish off, unfortunately, on a bit of a downer, and that is we're losing the very diversity that we're going to rely on. And we really are, some of you will have seen this in the EnviroHub talk a week or so ago, we really are facing the perfect storm in the marine domain from increased weather conditions, and that leads to catchment runoff, sediments in particular, pollutants come out, ocean acidification, marine invasions, all of these things happening at the same time, and that's equating to massive diversity loss. So the Waipu River, for instance, it's losing every year the equivalent of 80 dairy farms worth of topsoil. That's how much is going out. So it's no good for the farmers and certainly no good for the marine areas around it. So we've already lost, we know, large areas of marine biodiversity because of sediment inundation. It only takes a millimetre or two on the seabed to knock out those communities. And also we're losing our shallow water communities as well. So we're going from situations like this with kelp forests to situations like this because it's murky, light doesn't get through, so the plants aren't doing so well. So we've got some programs coming up to have a look at all of this. Okay, so that we are in a race against time. We absolutely are. We're wanting to preserve the diversity that we can then sample into the future for future generations. Looking to the future, there's all sorts of other things we can do, and it's fantastic being involved in drug discovery programs, but they will always be, given the nature of the cost, a consortium of approach involving big pharma. But we can do things which are more relevant to us quite quickly. And this is our concept, the blue to green. It's evoking the smart discovery ability to look at other useful bioactives. And how we can do that is because we have these databases. All the hard work has been done. We've made collections around the world, and we can now move up and down the sort of bioinformatics from the organisms, where they are, what they do, through to their chemistry, through to their modes of action, and then hunt out particular leads and particular sort of chemicals that we might be interested in. And here's an example. We knew that a number of marine organisms had Pseudomonas activity from, funnily enough, the human screens on the Pseudomonas lung condition. And we thought, when we learnt about the issue here with PSA and kiwi fruit, it's a Pseudomonas target, we thought, well, yeah, we know some things that can work there, particularly for the mode of action that Zespri were interested in, which was this quorum sensing modifying type activity where they are interested in something that will just turn off the pathogen. And so we went up to them and said, look, we think we have some leads for you might be interested in. They're novel. Shall we give it a go? And it's just based on the informatics. So we presented them with 5 targets, and all 5 came up active. And there's now one in particular that's being pursued through to on-farm trials very shortly. So we can map over our medicinal work into the agrochemical space and, of course, veterinary space and things like that. So we're in a pretty neat position, I think. Okay, COVID. Does the deep hold a remedy? If you Google COVID and New Zealand, you'll come up with an increasing number of links. And this is a really important one because there is a compound out there called glyphosate. That's this thing. And it's already been developed as an HIV/AIDS drug therapy, but its mode of action is quite relevant to SARS and COVID-19. So we suspect there's going to be probably a bit of interest in that. So there will be a Kiwi connection in our chase for effective COVID-19 antivirals. So coming back to this, anyone thought about how these connections might hook up? Well, there was an interesting observation. This is, once again, just a really simple example of if you look to nature, there are some answers out there. In some of the first trawls that we brought up of this sponge, we also brought up a number of these camouflage crabs. and they came from different trawls where the sponge wasn't, but they all ended up in the same sorting tray. And we noticed the camouflage crab was very quickly getting rid of bits of seaweed and other stuff, and it was going out to the sponge, the yellow slimy, Lissidendorix, the source of Halaven, and grabbing that and chopping it up and sticking it on its carapace instead. So it could recognise some chemical cues coming out of that sponge that it must have sensed, that's got toxins and stuff in there, I'm going to stick that on my back, I'm not going to be lunch for anybody. And that's what it did. So we actually ran a whole bunch of trials using camouflage crabs with sponges and various things we knew were active, and 100% they went out and they got exactly the right one. So this actually short-circuits the drug discovery program. We just need the odd camouflage crab. Anyway, I'll wind up now, just with these last few images. Some of you might have seen these before, but to me it just epitomises everything that this talk was about. I got sent these images from Shark Bay many years ago of dolphins swimming around with sponges on their noses. And, being a true scientist, we came up with a bunch of hypotheses because the researchers over in WA wanted to know what was going on. So here are the hypotheses. It could be using them as a game. It could be using the sponge for protection because the dolphins are known to go down on the bottom and snuffle around and scare up little fishes and things like that. It could be getting high on them. There's a lot of alkaloids and interesting compounds in there. Or it could be using them as a medicine. And it soon transpired, there's the sponge, echinodictyon, and it does have a lot of pretty interesting terpenes and other molecules in it. It turned out that the dolphins that were doing this were mainly pregnant females, and the suspicion is they're getting something out of that particular organism. The point I want to make here is that to make these connections, you need all of those elements in the food web, in the environment together, from the lowliest of sponges right the way through to the top end apex predators all together. And so that's what conservation biodiversity is all about, because our future really does rely on it. And in looking at these things, it's like the chimps, when they get sick, going to a particular plant, we can start to pick up on some clues and indeed look at some new therapies. So we're sitting in this country on an amazing biodiversity. It's unique. This image here, many of you, this is probably the first time you'll have seen this. This was taken, it's a bit grainy, it was just taken from a little GoPro type camera. It's at 50 metres, and it shows you a pretty amazing sponge garden just off Maititi Island. Okay, but you'll notice that things down the bottom there, there's really nothing, so because it's covered in sediments. So these are the refugia. Now there's a whole bunch of new species in here, and these organ pipe sponges and things you don't normally see south of the Poor Knights Islands. So we're sitting on a pretty unique spot right here. Really interesting. So we can harness some of the existing bioinformatics we have to be smarter and more accurate and faster in being able to find these leads. We can map them into spaces elsewhere that we can benefit as a country economically really quickly, and that's the agrochemical space. But we really are in a significant race against time. We really do need to try and halt this attrition of our seascapes, particularly from land inundation. So I'll leave it there, and thank you very much for your time coming.
Chris, thank you. Sponges, fascinating. Who would have thought? Ladies and gentlemen, traditionally we do finish now at 7 p.m, so if you need to leave, please feel free to do so. But we'll ask Chris to hang around for 10 minutes or so if you have questions. I think I'll leave the mic with Chris, so if you could raise your hand if you have a question and I point to you, if you could stand and project your voice, I'd appreciate that. So questions, please. If you need to leave, feel free to do that. Questions for Chris, please. Anybody? Yes, ma'am.
Do you think the sinking of the Reef in Tauranga Harbour, has that had an effect on the sponges or the other sea life that you can see substantially? Yeah, that's happened.
Okay, so the question was, has the RENA affected diversity in our ecosystem? No, it hasn't, which has been a good thing. We've been monitoring the effects of the RENA ever since it happened, and this October would be 10 years that reef is covered. It had an initial impact in the immediate zone, particularly where the ship hull was scraping. The legacy of it is probably associated with the tin coming off the hull, but the organisms have accommodated that. I think one thing the RENA showed us is just how quickly the marine environment can actually recover and how resilient it is. However, the RENA plus all sorts of other things probably wouldn't be a good thing. And we're very fortunate that the oil that was released from it was picked up very carefully by human hands on the beaches really, really quickly afterwards. If that hadn't happened, then yes, indeed, I think we would have seen a bit more of a legacy. Also, dispersants weren't used in that case, and we would have also seen a legacy of that because the Exxon Valdez, for instance, they're still seeing quite an impact in the shore life and around there. But in around Otaiti, Astrolabe Reef, you can dive. The RENA is there still. I mean, 2/3 of it, pretty much, down quite deep, but there's quite significant recovery in there. Many of the organisms will have taken up some of the metals. Whether they're edible or not is another thing you might wish to ask. But there appears to be no longer-term damage to the biodiversity out there, which is really good news.
Sir, you said that you had connections with China. Whoops. Connections with China. Tell me, the 3 universities there, what is the relationship, and do you grow and expand your knowledge because they're further ahead, or we're ahead?
The question is our relationship. What's the nature of our relationship with the agencies in China? The relationship's actually initially one of remediating damaged environments. And because the agencies, in Yantai in particular, lead the world in algal research. And they are charged primarily for looking at seaweeds that can actually enhance remediation of damaged environments. So up there, the Bohai Sea is effectively dead. And we are learning from them techniques to create cultivars of seaweeds that do good things, that can scrub up nutrients out of both fresh and marine waters and generate products that we can actually use, either as agri-feeds. Some of the species, for instance, you might have heard, if you feed them to cattle, it reduces their methane productions and emissions. Others can be used as soil amenities. So that's the prime use. However, the Institute of Materia Medica has a repository like ours. And so we're in a situation where we can share knowledge about where medicinal leads are, or agrochemical leads, or veterinary leads, and then look for the sources of those in this country, which are likely to be subtly different but potentially better. And so that's what we are doing, is sharing international databases. They are interested, I think, in the techniques that we're evolving, which is sort of looking at natural biosynthetic pathways to enhance the ability to actually create novel drug leads and so on. Yeah, good.
Yes, Chris, you mentioned the enormous filtering capacity of sponges earlier in your talk. Do the sponges actually pump the water, or just wait for it to go by? And the second half of my question, is there any future for using sponges for cleaning up the waterways that we are heavily polluting, or do they only live in the sea, in solid?
Yeah, good question. So the sponge pumping system is called an aquiferous system, and it's generated by a cell called a choanocyte, which is just a cell and it has a little flagella in it. And these choanocytes are grouped in chambers spread right throughout the sort of matrix of the sponge. There's a pretty neat YouTube clip, actually, you can watch. Probably don't have time to do that. But it takes you down inside a sponge, you know, in 3 dimensions, effectively, and then you get gobbled up by an archaeocyte, which comes and eats you because you... But yeah, these choanocytes just beat constantly, and they create a massive suction power with an exhalant canal, as I said, that runs at about 2 knots. So yeah, they're big pumping machines. We found out how efficient they were the hard way, though, rather sadly. We had a ton of water with a few sponges in the bottom, and we were transporting them into an aquarium system to grow them. This was the yellow slimy sponge to produce that drug. And they all died within an hour. And we had the presence of mind in the next time to put an oxygen probe in there. And so they stripped the oxygen out of the water column within half an hour out of a ton of water. And there's only, I don't know, a sprinkling of them. So their metabolism is huge. They just pump like crazy. Can you use them in a remediation situation? They're probably already doing that. The only issue with many of these lower invertebrates is that they can get smothered really easily. So the ones in Tauranga Harbour here, they have a mechanism that they can back flush every now and then and effectively wash off all the dust that settles on them, but many species can't. And so if you had them in muddy water, that would be a problem. But yeah, they could actually remediate other water sources as long as there wasn't too much sediment on it. It's interesting to note that both at the entrance of Tauranga main channel and also up near Bowentown are massive sponge communities that line the reef. It's quite deep out there, 30, 40 metres or so. But yeah, there's just sponge gardens and things, and they're acting as a massive filter for the harbour as well.
A few years back, we listened to a talk with reports that maybe the next antibiotics would come from the sea. You haven't talked about that today. Is there anything developing in that arena?
There will be for sure, but I'm not up to speed with the antibiotic program. The tumour active ones I am. I know that that group, you might have seen the acronym Pharmacy. We were the only Australasian partner on that. That's just coming to an end. It was funded, I think, by about 10 million euro. And they were directly looking for anti-infectives, antibiotics. They've come up with one lead in all that time. So you've actually hit the tricky territory in all of this. But having said that, I don't think anyone's applied some of the things that we perhaps have had into that therapeutic area. Maybe we should. It would be relatively simple to do so. Actually, we did run some experiments down the Antarctic where we inoculated them with a fungus that we saw growing down there, and there was an immediate response to that. So, yeah, you got me. I think we need to write our next Marsden grant on exactly that.
Thank you for your address tonight. This question is more about protection of the research team's findings. You mentioned Vesprit, so it comes to mind a massive recovery of our IP with thousands of acres of the gold kiwi now illegally being farmed.
Yep. No, absolutely relevant question, and the question was around IP protection. So we've actually, from this country, led the world in recognition of Indigenous rights, and we're fortunate in this country to have a treaty. It might interest you to know that you can't trade in any of the leads from marine or any other organism unless you have subscribed to the principles within the Convention on Biological Diversity. And there are two key things in that. One is mutually agreed terms with traditional owners where they might be known, and also
prior informed consent. So those two things have to be in place first, and then the protection can follow. So there should be no trade unless those sorts of things are invoked. In Australia, when I first went over there, because of the parlous state of Indigenous communities, Aboriginal and Torres Strait communities, many of them had lost knowledge of really where their rohi equivalent, if you like, was over there. And we initiated, actually it caused a bit of concern for the federal government there, a policy where, as you would in this country, you ask permission first before you go and sample anything, and you actually identify who the traditional owners are. And that's absolutely essential at the bottom step, because all of these other things that you're suggesting in terms of biopiracy of intellectual property and taking it away can then be safeguarded, because you have these international conventions to rely on. And the biggest one of those is this thing called the Nagoya Protocol. However, I don't think this country's signed up for that yet. And the reason it hasn't is because of the treaty settlement process hasn't been completed yet, but we aspire to those sorts of principles. Yes, sure. It's dire, to put it mildly. The Great Barrier Reef is suffering from the same sorts of situations that we're the perfect storm. Increasing frequency and intensity of cyclonic conditions, land runoff of enriched waterways from development of cane and sugar cane and banana and pineapple plantations, up and down the eastern seaboard of Queensland in particular. In addition to that, there's warming temperatures and a rising sea level and ocean acidification, all happening at the same time. So when you have acid seas, even just subtle shifts in the acid content of seawater, corals can't create skeleton anymore. And so they're eroded much more quickly. They can't grow to meet sort of these rising sea level conditions. With muddy waters coming out of many of the rivers through the Australian seaboard, as we have here, you have that turbidity where it's murky, they can't see. And what a lot of the inshore area of the Great Barrier Reef is now showing is what they call a phase shift, where it's moved from an acropora, coral-characterised ecosystem to one now characterised by seaweeds, so gas and seaweed. So that all of that diversity is lost. So Terry Hughes, who runs the ARC Centre of Excellence in Australia for looking at the Great Barrier Reef, says it's almost the end, because the prognosis in terms of carbon profiles and CO2 is now quite bad throughout Australia. There's a bit of a Tauranga connection here, not through me, but the current, well, he was one of my bosses for a while when I was at AIMS. His name's David Mead. He's the General Manager of AIMS, and he's been put in charge of a program designed to try and come up with some ways of ameliorating some of the demise and effects of climate change on the Great Barrier Reef. So there are people, for instance, who are looking at more resilient coral species. And corals, like many of the sponges that we're talking about, also have symbionts in them. That's what gives them their colour. They're called zooxanthellae, unicellular algae. And when it gets hot, the coral spits them out. If it doesn't cool down again soon enough, they'll die. But Madeleine Van Oppen, who's heading that up now, she's at AIMS and University of Melbourne, she's looking at creating clades of symbionts that are more robust and hardy, that they can sort of translocate into corals and use them, inoculate reefs and use them as a refugia. But really, it's a drop in the bucket with the issues there. If that wasn't enough, I can give you a talk on this. There's also crown of thorns starfish. And they, you know, like a lot of things that respond to bad conditions, they will come out in plague proportions, and there appears to be a relationship with their outbreaks with or periods of very intensive rainfall, because the larval life history phase is up in the estuaries, which benefits them. So they just come down in plagues, and what corals are left, they just come up and scour. So they're creating pretty unique ways of dealing with those now. They've got these underwater robots that can look and smell out crown of thorns and then inject them with formalin. They Crown of thorns, the acronym's COT, C-O-T, crown of thorns, and they call these things the COT bot. It just goes around they're murdering crown of thorns starfish. But it's, they're trying to do everything they possibly can to look at every, I guess, way they might be able to just chisel back a little bit of the resilience in the coral reefs. But we also, in this country, will be suffering from ocean acidification as well. We have a lot of coral species, more in the deeper water, but anything that needs a calcium carbonate skeleton in the sea is going to have issues. So shellfish, for instance. Yeah. Thank you.
Ladies and gentlemen, on your behalf, I'd like to thank Chris. Thank you, Chris, for sharing your passion for our oceans, for marine science and for sponges. Your knowledge is extraordinary, your enthusiasm is infectious, and your work ethic is second to none. You are so well respected and much loved across the university, Tauranga Moana and beyond. Ladies and gentlemen, Professor Chris Battersall, thank you.